Basal ganglia.
The basal ganglia comprise multiple subcortical nuclei, of varied origin, in the brains of vertebrates, which are situated at the base of the forebrain. Basal ganglia are strongly interconnected with the cerebral cortex, thalamus, and brainstem, as well as several other brain areas. The basal ganglia are associated with a variety of functions including control of voluntary motor movements, procedural learning, routine behaviors or habits such as bruxism, eye movements, cognition and emotion. Currently popular theories implicate the basal ganglia primarily in action selection. That is, it helps determine the decision of which of several possible behaviors to execute at any given time. In more specific terms, the basal ganglia's primary function is likely to control and regulate activities of the motor and premotor cortical areas so that voluntary movements can be performed smoothly. Experimental studies show that the basal ganglia exert an inhibitory influence on a number of motor systems, and that a release of this inhibition permits a motor system to become active. The behavior switching that takes place within the basal ganglia is influenced by signals from many parts of the brain, including the frontal cortex, which plays a key role in executive functions. The main components of the basal ganglia are the striatum, caudate nucleus and putamen, the globus pallidus, the substantia nigra, the nucleus accumbens, and the subthalamic nucleus. Each of these areas has a complex internal anatomical and neurochemical organization. The largest component, the striatum, receives input from many brain areas beyond the basal ganglia, but only sends output to other components of the basal ganglia. The pallidum receives input from the striatum, and sends inhibitory output to a number of motor-related areas. The substantia nigra is the source of the striatal input of the neurotransmitter dopamine, which plays an important role in basal ganglia function. The subthalamic nucleus receives input mainly from the striatum and cerebral cortex, and projects to the globus pallidus. The importance of these subcortical nuclei for normal brain function and behavior is emphasized by the numerous and diverse neurological conditions associated with basal ganglia dysfunction, which include disorders of behavior control such as Tourette syndrome, hemiblismus, and obsessive-compulsive disorder, dystonia, psychostimulant addiction, and movement disorders, the most notable of which are Parkinson's disease, which involves degeneration of the dopamine-producing cells in the substantia nigra pars compacta, and Huntington's disease, which primarily involves damage to the striatum. The basal ganglia have a limbic sector whose components are assigned distinct names, the nucleus accumbens, ventral pallidum, and ventral tegmental area, VTA. There is considerable evidence that this limbic part plays a central role in reward learning, particularly a pathway from the VTA to the nucleus accumbens that uses the neurotransmitter dopamine. A number of highly addictive drugs, including cocaine, amphetamine, and nicotine, are thought to work by increasing the efficacy of this dopamine signal. There is also evidence implicating overactivity of the VTA dopaminergic projection in schizophrenia.
Structure.
In terms of development, the human nervous system is often classified based on the original three primitive vesicles from which it develops. These primary vesicles form in the normal development of the neural tube of the human fetus and initially include prosencephalon, mesencephalon, and rhombencephalon, in rostral to caudal, from head to tail, orientation. Later in development of the nervous system each section itself turns into smaller components. Mr. Virus sometimes loses his amygdala experience hypertatification, hyperality and hypersexuality. During development, the cells that migrate tangentially to form the basal ganglia are directed by the lateral and medial ganglionic eminences. The following table demonstrates this developmental classification and traces it to the anatomic structures found in the basal ganglia. The structures relevant to the basal ganglia are shown in bold. The basal ganglia form a fundamental component of the cerebrum. In contrast to the cortical layer that lines the surface of the forebrain, the basal ganglia are a collection of distinct masses of gray matter lying deep in the brain not far from the junction of the thalamus. Like most parts of the brain, The basal ganglia consist of left and right sides that are virtual mirror images of each other. In terms of anatomy, the basal ganglia are divided by anatomists into 4 distinct structures, depending on how superior or rostral they are, in other words depending on how close to the top of the head they are. 2 of them, the striatum and the pallidum, are relatively large. The other 2, the and the subthalamic nucleus, are smaller. In the illustration to the right, 2 coronal sections of the human brain show the location of the basal ganglia components. Of note, and not seen in this section, the subthalamic nucleus and substantia nigra lie further back, posteriorly, in the brain than the striatum and pallidum.
Striatum.
The striatum is the largest component of the basal ganglia. The term striatum comes from the observation that this structure has a striped appearance when sliced in certain directions, arising from numerous large and small bundles of nerve fibers, white matter, that traverse it. Early anatomists, examining the human brain, perceived the striatum as 2 distinct masses of gray matter separated by a large tract of white matter called the internal capsule. They named these 2 masses the caudate nucleus, and putamen. More recent anatomists have concluded, on the basis of microscopic and neurochemical studies, that it is more appropriate to consider these masses as 2 separated parts of a single entity, the striatum, in the same way that a city may be separated into 2 parts by a river. Numerous functional differences between the caudate and putamen have been identified, but these are taken to be consequences of the fact that each sector of the striatum is preferentially connected to specific parts of the cerebral cortex. The internal organization of the striatum is extraordinarily complex. The great majority of neurons, about 96%, are of a type called medium spiny neurons. These are GABAergic cells, meaning that they inhibit their targets, with small cell bodies and dendrites densely covered with dendritic spines, which receive synaptic input primarily from the cortex and thalamus. Medium spiny neurons can be divided into subtypes in a number of ways, on the basis of neurochemistry and connectivity. The next most numerous type, around 2%, are a class of large cholinergic interneurons with smooth dendrites. There are also several other types of interneurons making up smaller fractions of the neural population. Numerous studies have shown that the connections between cortex and striatum are, in general, topographic. That is, each part of the cortex sends stronger input to some parts of the striatum than to others. The nature of the topography has been difficult to understand, however, perhaps in part because the striatum is organized in 3 dimensions, whereas the cortex, as a layered structure, is organized in 2. This dimensional discrepancy entails a great deal of distortion and discontinuity in mapping one structure to the other. It is interesting to note that the same topography applies to the striatal connections to the thalamus.
Pallidum.
The pallidum consists of a large structure called the globus pallidus, pale globe, together with a smaller ventral extension called the ventral pallidum. The globus pallidus appears as a single neural mass, but can be divided into 2 functionally distinct parts, called the internal, or medial, and external, lateral, segments, abbreviated GPI and GPE. Both segments contain primarily GABAergic neurons, which therefore have inhibitory effects on their targets. The 2 segments participate in distinct neural circuits. The external segment, or GPE, receives input mainly from the striatum, and projects to the subthalamic nucleus. The internal segment, or GPI, receives signals from the striatum via 2 pathways, called direct and indirect. Pallidal neurons operate using a disinhibition principle. These neurons fire at steady high rates in the absence of input, and signals from the striatum cause them to pause or reduce their rate of firing. Because pallidal neurons themselves have inhibitory effects on their targets, the net effect of striatal input to the pallidum is a reduction of the tonic inhibition exerted by pallidal cells on their targets, disinhibition, with an increased rate of firing in the targets.
Substantia nigra.
The substantia nigra is a mesencephalic gray matter portion of the basal ganglia that is divided into SNR, reticulator, and SNC, compactor. SNR often works in unison with GPI, and the SNR-GPI complex inhibits the thalamus. Substantia nigra pars compactor, SNC, however, produces the neurotransmitter dopamine, which is very significant in maintaining balance in the striatal pathway. The circuit portion below explains the role and circuit connections of each of the components of the basal ganglia.
Subthalamic nucleus.
The subthalamic nucleus, STN, is a diencephalic gray matter portion of the basal ganglia, and the only portion of the ganglia that produces an excitatory neurotransmitter, glutamate. The role of the subthalamic nucleus is to stimulate the SNR-GPI complex, and it is part of the indirect pathway. The subthalamic nucleus receives inhibitory input from the external part of the globus pallidus and sends excitatory input to the GPI.
Circuit connections.
In order to understand the complex circuitry of the basal ganglia, one has to first understand the important participants in this circuit. Parts of the basal ganglia are in direct communication with the thalamus and the cortex. The cortex, thalamus, and the basal ganglia are, therefore, the three main participants in the circuit created by the basal ganglia. At the top of the hierarchy lies the cerebral cortex. The cortex has many different areas with different functions. One such cortical area is called the primary motor cortex, along the precentral gyrus. Specialized neurons from the primary motor cortex extend their axons all the way to the striatum portion of the basal ganglia. These cortical neurons release the neurotransmitter glutamate, which is excitatory in nature. Once excited by glutamate, the cells in the striatum problem in two different directions, giving rise to two major pathways, the direct and the indirect pathways. In the direct pathway, cortical cells project excitatory inputs to the striatum, which in turn projects inhibitory neurons onto the cells of the SNR-GPI complex. The SNR-GPI complex projects directly onto the thalamus through the inhibitory answer lenticularis pathway. The striatal inhibition of the SNR-GPI complex coupled with SNR-GPI inhibition of the thalamus therefore results in a net reduction of inhibition of the thalamus via the striatum. The thalamus projects excitatory glutamatergic neurons to the cortex itself. The direct pathway, therefore, results in the excitation of the motor cortex by the thalamus. Once stimulated, the cortex projects its own excitatory outputs to the brainstem and ultimately muscle fibers via the lateral corticospinal tract. The following diagram depicts the direct pathway. The indirect pathway also starts from neurons in the striatum. Once stimulated by the cortex, striatal neurons in the indirect pathway project inhibitory axons onto the cells of the globus pallidus externa, GPE, which tonically inhibits the subthalamic nucleus, STN. This inhibition, by the striatum, of the inhibitory projections of the GPE, results in the net reduction of inhibition of the STN. The station, in turn, projects excitatory inputs to the SNR-GPI complex, which inhibits the thalamus. The end result is inhibition of the thalamus and, therefore, decreased stimulation of the motor cortex by the thalamus and reduced muscle activity. The direct and indirect pathways are therefore antagonist in their functions. Following is a diagram of the indirect pathway. The antagonistic functions of the direct and indirect pathways are modulated by the Substantia Nigra pars compactor, SNC, which produces dopamine. In the presence of dopamine, D1 receptors in the basal ganglia stimulate the GABAergic neurons, favoring the direct pathway, and thus increasing movement. The GABAergic neurons of the indirect pathway are stimulated by excitatory neurotransmitters acetylcholine and glutamate. This sets off the indirect pathway that ultimately results in inhibition of upper motor neurons, and less movement. In the presence of dopamine, D2 receptors in the basal ganglia inhibit these GABAergic neurons, which reduces the indirect pathway's inhibitory effect. Dopamine therefore increases the excitatory effect of the direct pathway, causing movement, and reduces the inhibitory effect of the indirect pathway, preventing full inhibition of movement. Through these mechanisms the body is able to maintain balance between excitation and inhibition of motion. Lack of balance in this delicate system leads to pathologies such as Parkinson's disease. Parkinson's disease involves the loss of dopamine, which means the direct pathway is less able to function, so no movement is initiated, and the indirect pathway is in overdrive, causing too much inhibition of movement.
Function.
Information about the functions of the basal ganglia comes from anatomical studies, from physiological studies carried out mainly in rats and monkeys, and from the study of diseases that damage them. The greatest source of insight into the functions of the basal ganglia has come from the study of two neurological disorders, Parkinson's disease and Huntington's disease. For both of these disorders, the nature of the neural damage is well understood and can be correlated with the resulting symptoms. Parkinson's disease involves major loss of dopaminergic cells in the substantia nigra. Huntington's disease involves massive loss of medium spiny neurons in the striatum. The symptoms of the two diseases are virtually opposite. Parkinson's disease is characterized by gradual loss of the ability to initiate movement, whereas Huntington's disease is characterized by an inability to prevent parts of the body from moving unintentionally. It is noteworthy that, although both diseases have cognitive symptoms, especially in their advanced stages, the most salient symptoms relate to the ability to initiate and control movement. Thus, both are classified primarily as movement disorders. A different movement disorder, called hemiballismus, may result from damage restricted to the subthalamic nucleus. Hemiballismus is characterized by violent and uncontrollable flinging movements of the arms and legs.
Eye movements.
One of the most intensively studied functions of the basal ganglia, BG, is their role in controlling eye movements. Eye movement is influenced by an extensive network of brain regions that converge on a midbrain area called the superior colliculus, SC. The SC is a layered structure whose layers form two-dimensional retinotopic maps of visual space. A bump of neural activity in the deep layers of the SC drives an eye movement directed toward the corresponding point in space. The SC receives a strong inhibitory projection from the BG, originating in the substantia nigra pars reticulata, SNR. Neurons in the SNR usually fire continuously at high rates, but at the onset of an eye movement they pause, thereby releasing the SC from inhibition. Eye movements of all types are associated with pausing in the SNR. However, individual SNR neurons may be more strongly associated with some types of movements than others. Neurons in some parts of the caudate nucleus also show activity related to eye movements. Since the great majority of caudate cells fire at very low rates, this activity almost always shows up as an increase in firing rate. Thus, eye movements begin with activation in the caudate nucleus, which inhibits the SNR via the direct GABAergic projections, which in turn disinhibits the SC.
Role and motivation.
Although the role of the basal ganglia in motor control is clear, there are also many indications that it is involved in the control of behavior in a more fundamental way, at the level of motivation. In Parkinson's disease, the ability to execute the components of movement is not greatly affected, but motivational factors such as hunger fail to cause movements to be initiated or switched at the proper times. The immobility of Parkinsonian patients has sometimes been described as a paralysis of the will. These patients have occasionally been observed to show a phenomenon called kinesia paradoxica, in which a person who is otherwise immobile responds to an emergency in a coordinated and energetic way, then lapses back into immobility once the emergency has passed. The role in motivation of the limbic part of the basal ganglia, the nucleus accumbens, NA, ventral pallidum, and ventral tegmental area, VTA, is particularly well established. Thousands of experimental studies combine to demonstrate that the dopaminergic projection from the VTA to the NA plays a central role in the brain's reward system. Animals with stimulating electrodes implanted along this pathway will bar press very energetically if each press is followed by a brief pulse of electrical current. numerous things that people find rewarding, including addictive drugs, good-tasting food, and sex, have been shown to elicit activation of the VTA dopamine system. Damage to the NA or VTA can produce a state of profound torpor. Although it is not universally accepted, some theorists have proposed a distinction between appetitive behaviors, which are initiated by the basal ganglia, and consummatory behaviors, which are not. For example, an animal with severe basal ganglia damage will not move toward food even if it is placed a few inches away, but, if the food is placed directly in the mouth, the animal will chew it and swallow it.
Neurotransmitters.
In most regions of the brain, the predominant classes of neurons use glutamate as neurotransmitter and have excitatory effects on their targets. In the basal ganglia, however, the great majority of neurons use GABA as neurotransmitter and have inhibitory effects on their targets. The inputs from the cortex and thalamus to the striatum and STN are glutamatergic, but the outputs from the striatum, pallidum, and substantia nigra pars reticulata all use GABA. Thus, following the initial excitation of the striatum, the internal dynamics of the basal ganglia are dominated by inhibition and disinhibition. Other neurotransmitters have important modulatory effects. The most intensively studied is dopamine, which is used by the projection from the substantia nigra pars compacta to the striatum, and also in the analogous projection from the ventral tegmental area to the nucleus accumbens. Acetylcholine also plays an important role, being used both by several external inputs to the striatum, and by a group of striatal interneurons. Although cholinergic cells make up only a small fraction of the total population, the striatum has one of the highest acetylcholine concentrations of any brain structure.
Clinical significance.
The following is a list of disorders that have been linked to the basal ganglia. Athetosis, Athymic syndrome, PAP syndrome, attention deficit hyperactivity disorder, ADHD, blepharospasm, bruxism, cerebral palsy, basal ganglia damage during second and third trimester of pregnancy, chorea, dystonia, Fahrenheit's disease, foreign accent syndrome, FAS, Huntington's disease, kernicterus, Lesch-Nyhan syndrome, major depressive disorder, obsessive-compulsive disorder, other anxiety disorders, PANDAS, Parkinson's disease, spasmodic dysphonia, stuttering, Sydenham's chorea, tardive dyskinesia, caused by chronic antipsychotic treatment, Tourette's disorder, Wilson's disease.
History.
The acceptance that the basal ganglia system constitutes one major cerebral system took time to arise. The first anatomical identification of distinct subcortical structures was published by Thomas Willis in 1664. For many years, the term corpus striatum was used to describe a large group of subcortical elements, some of which were later discovered to be functionally unrelated. For many years, the putamen and the caudate nucleus were not associated with each other. Instead, the putamen was associated with the pallidum in what was called the nucleus lenticularis or nucleus lentiformis. A thorough reconsideration by Cecile and Oscar Vogt, 1941, simplified the description of the basal ganglia by proposing the term striatum to describe the group of structures consisting of the caudate nucleus, the putamen, and the mass linking them ventrally, the nucleus accumbens. The striatum was named on the basis of the striated, striped, appearance created by radiating dense bundles of striatopallidonigral axons, described by anatomist Samuel Alexander Kinnear Wilson, 1912, as pencil-like. The anatomical link of the striatum with its primary targets, the pallidum and the substantia nigra, was discovered later. The name Globus Pallidus was attributed by Degerine to Bowditch, 1822. For this, the Vogts proposed the simpler pallidum. The term locus niger was introduced by Félix Victor as tache noire in 1786, though that structure has since become known as the substantia nigra, due to contributions by von Summering in 1788. The structural similarity between the substantia nigra and Globus Pallidus was noted by Murto in 1896. Together, the two are known as the pallidonigral ensemble, which represents the core of the basal ganglia. Altogether, the main structures of the basal ganglia are linked to each other by the striato-pallidonigral bundle, which passes through the pallidum, crosses the internal capsule as the comb bundle of Edinger, then finally reaches the substantia nigra. Additional structures that later became associated with the basal ganglia are the body of Lewis, 1865, nucleus of Lewis on the figure, or subthalamic nucleus, whose lesion was known to produce movement disorders. More recently, other areas such as the central complex, center median parafascicular, and the peduncular-pontine complex have been thought to be regulators of the basal ganglia. Near the beginning of the 20th century, the basal ganglia system was first associated with motor functions, as lesions of these areas would often result in disordered movement in humans, chorea, athetosis, Parkinson's disease.
Terminology.
The nomenclature of the basal ganglia system and its components has always been problematic. Early anatomists, seeing the macroscopic anatomical structure but knowing nothing of the cellular architecture or neurochemistry, grouped together components that are now believed to have distinct functions, such as the internal and external segments of the globus pallidus, and gave distinct names to components that are now thought to be functionally parts of a single structure, such as the caudate nucleus and putamen. The term basal comes from the fact that most of his elements are located in the basal part of the forebrain. The term ganglia is a misnomer. In modern usage, neural clusters are called ganglia only in the peripheral nervous system. In the central nervous system they are called nuclei. For this reason, the basal ganglia are also occasionally known as the basal nuclei. Terminologia Anatomica, 1998, the International Authority for Anatomical Naming, retained nuclei basals, but this is not commonly used. The International Basal Ganglia Society, IBAGS, informally considers the basal ganglia to be made up of the striatum, the pallidum, with two nuclei, the substantia nigra, with its two distinct parts, and the subthalamic nucleus. Percheronale in 1991 and Parent in 2005 included the central region, center median parafascicular, of the thalamus as part of the basal ganglia, while Minosegovia al. in 2004 included the peduncular-pontine complex as well. Also, the names given to the various nuclei of the basal ganglia are different in different species. In particular, the internal segment of the globus pallidus in primates is called the entopeduncular nucleus in rodents. The striatum, and external segment of the globus pallidus in primates are called the paleostriatum augmentatum, and paleostriatum primitivum, respectively, in birds.
In other animals.
The basal ganglia form one of the basic components of the forebrain, and can be recognized in all species of vertebrates. Even in the lamprey, generally considered one of the most primitive of vertebrates, striatal, pallidal, and nigral elements can be identified on the basis of anatomy and histochemistry. A clear emergent issue in comparative anatomy of the basal ganglia is the development of this system through phylogeny as a convergent cortically reentrant loop in conjunction with the development and expansion of the cortical mantle. There is controversy, however, regarding the extent to which convergent selective processing occurs versus segregated parallel processing within reentrant closed loops of the basal ganglia. Regardless, the transformation of the basal ganglia into a cortically reentrant system in mammalian evolution occurs through a redirection of pallidal, or paleostriatum primitivum output from midbrain targets such as the superior colliculus, as occurs in sauropsid brain, to specific regions of the ventral thalamus and from there back to specified regions of the cerebral cortex that form a subset of those cortical regions projecting into the striatum. The abrupt rostral redirection of the pathway from the internal segment of the globus pallidus into the ventral thalamus, via the path of the ansa lenticularis, could be viewed as a footprint of this evolutionary transformation of basal ganglia outflow and targeted influence.